We're going to take a look at DNA replication in this lecture. There's actually two ways that genetic information can flow in cells. So we have the central dogma and also vertical gene transfer, and we've talked about both of these before. So remember that the central dogma or the central principle of biology is just going from the gene in the DNA and transcribing that into the messenger RNA, and then we'll use the ribosome to translate the messenger RNA. So we'll read the code and then make that protein coded for by the gene and the DNA. So that's our central dogma, and that's what's pictured over here on the right. Okay, and then we also have vertical gene transfer. And remember, vertical gene transfer is just where the cell is reproducing. And so maybe we've got this bacterial cell here, its chromosome, and when it goes to divide, it's just going to make a copy of its chromosome. And then each of its daughter cells will get a copy of that chromosome. And so they call it vertical gene transfer because the genetic information is being passed from one generation down to the next generation. So it's vertical because it's going down the generation. So in this picture over here on the right, vertical gene transfer is shown here on the left. All right, so let's take a look at DNA replication. So this will be looking at this vertical gene transfer. So the cell will be reproducing. So the reason that the cell is going to replicate its DNA, why it would want to make a copy of its chromosome, is that the cell is getting ready to divide. So that's kind of the big picture here. So don't forget about that. Because you need to make sure that each of the daughter cells will get a copy of each chromosome. Okay, so we've got one chromosome that is pictured here, and we're going to make a copy of it. So we're just going to take the double-stranded DNA and we're just going to break the hydrogen bonds that are holding the two DNA strands together. And then we're going to use each of these old parent strands that are in blue as a template, and we're going to make a complementary new strand to go with each of the old parent strands. So the new strand will be here in purple and the old parent strand will be here in blue. So we're going to say that synthesis is semi-conservative. Okay, semi meaning half, and conserving just meaning that we're going to conserve half of the old strand, and then half of this will be new. So this chromosome that we've copied here, half of it's going to be new and half of it will be old. So that's what we mean by semi-conservative. All right, let's take a look at DNA replication in a bacterial cell, which is pretty easy because they just have one chromosome to copy, and it's circular. So they're going to start replicating their chromosome at what's called the origin of replication. So there's just a sequence in the DNA that the enzymes that are going to do the DNA replication will recognize and say, okay, that's where we need to break the hydrogen bonds, separate the DNA strands there, and then the enzymes are going to start going around both directions. So the DNA replication is going to be bidirectional, so it's going to go both ways around the circle, and they will just go around until they meet on the other side, and there's a termination sequence here that says, hey, that's the end of the replication, that's the end of the chromosome that you have to replicate, and then the enzymes will stop replicating and fall off. Okay, and then they will just separate the two chromosomes apart, and then they will separate those into the two daughter cells. And then they're done with their binary fission, so pretty easy. Okay, DNA replication in eukaryotes, we've got a little bit more going on. So here we're just showing one of the linear eukaryotic chromosomes. And remember, we would have two or more chromosomes in a eukaryotic cell, depending on the species. And if we take a look at this picture, how is the DNA replication going to be different from bacterial DNA replication? So looking at this, hopefully you noticed that there's several places that we've started replicating the DNA here. So there's actually many origins of replication. There's not just one like we had with the bacterial chromosome, but there's many. And we're going to have the enzymes that are replicating the DNA. They will start working in both directions, making the new DNA strands to go with the old parent strand. And then these replication bubbles will then just fuse together. And now we've got two chromosomes where we just started with one. And each of these chromosomes is half old and half new. Okay, that's that semi-conservative replication. So how the DNA replication is different from the bacteria, we have many origins of replication, whereas the bacteria just had one. And the reason that eukaryotes have many origins of replication is if you think about how much DNA they have compared to a bacterial cell, they've got a lot more, right? They've got two or more chromosomes that they've got to copy, and so if you only had one origin of replication per chromosome, that would take you a long time to copy all those chromosomes. So it speeds it up if you have many origins of replication on each chromosome. All right, there's a lot of different enzymes that are involved in DNA replication. I know that you've learned these before, so let's just review these. So we'll go through and actually look at a picture of these. I think that'll be a little bit easier, but they're all written down here what they do. And then let's just answer this question down here on the bottom, just kind of reminding ourselves just to kind of tie this in with the antibiotics that we just learned about. So remember, there was an antibiotic that kills bacteria by inhibiting the enzyme topoisomerase. And topoisomerase is going to undo all the knotting and the twisting in the DNA during DNA replication. So hopefully you remembered that the antibiotic that inhibits topoisomerase, those were the quinolones. Okay, that inhibits the topoisomerase. All right, and then just filling in some more blanks here. The enzyme that's actually going to make the new strand of DNA is called DNA polymerase III. And it's going to be reading that parent template strand in the 3' to 5' direction. And then it's going to be synthesizing the new strand of DNA in the 5' to 3' direction. So I would memorize that. That probably would make a great test question. So I would know those directions. So if you have your DNA here and you've got your template parent strand here, and maybe you've got, here's the sequence. So there's your DNA template strand that you're going to be reading in the 3 to 5 prime direction. And then the new strand that you're going to make to go with this old parent strand is going to be made in the 5 to 3 prime direction. Okay, and so remember the two DNA strands are always anti-parallel to each other. They always run in the opposite directions, and they're always going to be complementary. So if you remember apple tree and chewing gum, that will help you keep those straight. So A's and T's, C's and G's always go together. Okay, so the reason that this new strand can only be made in the 5 to 3 prime direction is because the 3 prime end has a hydroxyl group on it, whereas the 5 prime end has a phosphate group on it. And you can only add new nucleotides to this 3 prime end where there's this hydroxyl group, because that's pretty easy to chop off. But you can't add nucleotides to the 5 prime end because it's really hard to chop that phosphate group off. Okay, so you can never add to the 5 prime end, so you can only add to that 3 prime end. So that's why we have the directions that we do. So just memorize that you always have to read in the 3 to 5 prime direction, and we're going to be synthesizing or making the new DNA strand in the 5 to 3 prime direction. So just memorize that. Okay, and then taking a look at this picture, we've got our old parent template strand here on the right. And then here's the new strand that we're making. And you can see our 3 prime end is right here with the hydroxyl group. And here you can see our next nucleotide that we're going to be adding in here. We've got a T to go with that A on the old strand. And answering our questions here, is this an anabolic or a catabolic reaction? So we're making this new strand and we're making it bigger, right? We're making it longer. So this would be an anabolic reaction, right? Because we're making a larger molecule. And because this is an anabolic reaction, this is an endergonic uphill reaction. We've got to put energy in to get this reaction to go. So looking at this picture, do you see anything here that would supply the energy to make this reaction go, to drive it up the hill? Okay, so hopefully you notice that the nucleotide here has 3 phosphate groups, right? And what we're going to do is we're going to chop 2 of them off. And these covalent bonds that are holding the phosphate groups on, they store a lot of energy. So when you chop those phosphate groups off, that releases a lot of energy. So that's where the energy comes from to drive this reaction. Okay. All right, and then also during DNA replication, we have to deal with a leading strand and a lagging strand. So let's look at a picture of this. So here we've got our old parent template strand in the dark blue, and then we've got the new strand here in the light blue. Okay, and we're just going to look at 1/2 here of the replication bubble. And this top light blue strand, our new strand that we're making here, this is our leading strand. So notice that it's being made going towards that replication fork, and it's just being made in one long continuous piece. Okay, so that's your leading strand. The lagging strand, Okay, they call it that because it lags behind. It takes a little bit longer to make. And it's going to be made in several small pieces called Okazaki fragments, just named after the scientist that discovered this. So the first Okazaki fragment is actually right here. Okay, and then our second Okazaki fragment we made is right here, and then our third one is right here. So we're going to make our first Okazaki fragment going away from the replication fork. And then we're going to be backing up, and then we'll make our second Okazaki fragment, again going away from the replication fork. Oops, the little pointer here is not working too well. And then we'll back up and make our third Okazaki fragment, again going away from the replication fork. Okay, so that's the lagging strand. All right, and then we'll talk about all of these enzymes on our next picture here. All right, let's take a look at this picture that has all of the enzymes used in DNA replication on it. And there's also a nice tutorial animation that also shows DNA replication as well if you want to watch that at home. So this link is here on the bottom. And we're actually going to start over here on the far left. And we actually don't have in this picture, but the topoisomerase enzyme would be running here ahead of the helicase, which is in green. So this is topoisomerase. So topoisomerase's job is to undo all the knotting and the twisting in the DNA. Okay, and then we've got helicase here in green, and helicase is going to break the hydrogen bonds and separate the two DNA strands apart. Okay, and then to keep the DNA strands from going back together, we have these gray things here. These are the single-stranded binding proteins. Okay, and there's some up here too, so they'll be on both sides. Okay, so that'll keep the two DNA strands from joining back together because we need to keep them apart while we make the new strands here. All right, so now that we've got the two DNA parent strands apart, now we can use them as templates to make our new strands to go along with the old parent strands. But we need a primer to start the new strands. So we're going to use the enzyme called primase, which is actually pictured over here in pink. But we're going to start over here on the top, and the primase enzyme is going to make the primer here in red, and the primer is actually going to be made out of RNA. Although we are making a DNA strand, the primer is made out of RNA, which we'll replace with DNA a little bit later. Okay, and now we have something to start with, and then DNA polymerase 3, which is actually all the way down here right now, will come in and now start adding the DNA nucleotides to make our new DNA strand. Okay, and this will be our leading strand because it's just being made in one long continuous piece. Okay. All right, so that top one is pretty easy. Now the bottom one, got a little bit more going on. This one's going to be our lagging strand. So we actually started with our first Okazaki fragment down here, and then we did our second one, third, fourth, and then here's our most recent Okazaki fragment that we're starting with our primase enzyme making the RNA primer. And then once we have our RNA primer, then DNA polymerase III will come in and put in the DNA nucleotides until it runs into the previous RNA primer, and then it falls off. And then we need to get rid of the RNA from the primer and replace it with DNA. So DNA polymerase I will come in and replace the RNA primer with DNA nucleotides. So that's what DNA polymerase I is doing here. So it's just taking out the RNA nucleotides and replacing them with DNA. But it can't join the Okazaki fragments together. It leaves a little gap there. So we have another enzyme called DNA ligase that will come in and join the Okazaki fragments together. Okay, so if you remember, the word ligate means to tie together. That might help you remember that enzyme. Okay, so I would definitely know all of these enzymes and what they do, because I could very easily, and I will, put this on the quiz and the exam and ask you what these enzymes are doing. Okay, so make sure you know these, because these are just pretty easy to get right on the quiz and the exam if you know what they do. Okay. All right, and actually all of these replication enzymes are joined together into a little replicating machine called a replisome. Zome just means body or structure. Okay. All right, and then this is just showing you in the bacteria where we've got the origin of replication, where they start replicating the DNA, going around both sides, and then they meet at the termination site, a special sequence on the DNA that just says, hey, we're done copying the DNA replication, that's it. And so the enzymes just fall off, and then they just separate the two chromosomes, and then they just finish dividing those into the two daughter cells. Okay, it's a little bit different in eukaryotic cells because they have linear chromosomes. They're not circular. And remember that the lagging strand, we can't finish replicating that. And so every time you replicate your chromosomes, the ends of the chromosomes get shorter. And so what we do with our chromosomes is we have these telomeres. So these telomeres are just the ends of the eukaryotic chromosomes that contain just these non-coding repetitive sequences of DNA. They don't code for any genes, because we know that we can't finish replicating those, and they're going to get shorter each time we replicate them, and we're going to end up losing parts of our telomeres. So they don't contain any genes. Okay, so those are our telomeres. And you can see in this picture here, on this telomere, this end part of this chromosome, you can see that this part here, this lagging strand, it wasn't finished. So you can see how it's shortened. Okay, but there are some cells that divide a lot, and so they would very quickly run out of telomeres and then start losing parts of their genes. So we have an enzyme called telomerase that will actually lengthen the telomeres. And so in certain cells in our body that divide a lot, they've got this telomerase gene turned on, and they make this enzyme to lengthen their telomeres. And so this is just showing you how this enzyme works, and it will lengthen the longer strand here, and that gives room to put a primer on, and then we can finish filling in the DNA nucleotides and then make that telomere longer, and then the cell can keep dividing longer. So for example, skin cells, your intestinal cells, they have telomerase that's active. Okay, so let's fill in this table. So this would be good to study. So differences in DNA replication between bacteria and eukaryotes. So the number and shape of chromosomes, we have one circular chromosome in bacteria. And then the eukaryotes have 2 or more linear chromosomes. And the number of origins of replication per chromosome for bacteria, they just have 1. And then eukaryotes will have many origins of replication on each chromosome. And then as far as the rate of DNA replication, bacteria are going to be pretty fast. They're going to be very quick at replicating their DNA, whereas eukaryotes are going to be slower because they just have a lot more DNA to replicate. And bacteria do not have any telomerase because they don't have any telomeres. Their chromosome is circular. They don't have any ends to their chromosomes. And eukaryotes do have telomeres and they do have telomerase. And then just the last thing, plasmids that we find in prokaryotic cells, and there are a few in some eukaryotic cells like some yeast. Plasmids do replicate as well. So they replicate a little bit differently though. They're interesting. So what they do is they're double-stranded. So they've got the 2 strands of DNA. And what happens is one strand will get a nick in it. So it'll break. And then the one strand just starts pulling off the circle. And then where it leaves, the single strand that's left acts as a template and we'll make a new strand to go with it. Okay, so it just goes around. So we call it a rolling circle replication because it just rolls around and makes a new strand to go where the old strand had been pulled off. And then this old strand that gets pulled off, then you can just make a new strand to go with that one too. Okay, and then you can pass that on to the daughter cells as well.